Hello to everyone. Welcome to our webinar. I'm welcoming all our speakers and all our attendees. I'm Alexander Markovic from MDPI, and I will just provide a brief introduction to today's webinar, and later I will pass the word to our speakers. I will briefly introduce MDPI to those who maybe have not collaborated with us in the past, but I really hope that in future we will collaborate. And you probably know that MDPI is one of pioneer in open access publishing. We've been active since 1996 when we were founded by our founder, Dr. Shukun Lin. Our headquarters are in Basel, and since then we are fostering open scientific exchange in all forms across various disciplines. So far, we have 478 diverse and open access journals, including 469 peer-reviewed journals and 9 conference journals. And we are supported by almost 300,000 academic experts who are sharing our mission, values, and commitment to provide high-quality service for all authors. Today's topic, today's webinar is aiming to promote one of our topic that collects papers in genetic engineering in agriculture that focus on genetic engineering application in agriculture, including crops, livestock, breeding, and some other fields. The topic is edited by Dr. Amy Coco from University of Colorado, and she is leading this topic together with co-guest editors Dr. Chen from University of Florida and Dr. Lu from Central Community College, Hastings. We already published first edition of topic that collected 11 papers, and now we are running a second edition of this topic. And we will share the link that can lead you to the topic web page. So if you are interested in submission, you can submit your papers to the topic. Here is the program of today's webinar. At the beginning, our chair Dr. Coco will give introduction speech, and also she will present her colleagues who will provide presentations regarding that are connected with their research. Our speakers for today webinars are Dr. Bernard Won, Miss Ivy Lind, Mr. Sebastian Koisoba, and after them I will also give a word again to our chair, and after that the meeting and the webinar will be closed. I encourage you to leave your questions in our Q&A section, and each of speaker will check the Q&A section after they finish with their presentations, and we will gladly provide answers to your questions. Also, I encourage you that at the end of the webinar, you can leave general questions that may be connected to the whole field, not to the just specific presentation. So now I will invite our chair, Dr. Amy Coco, to start with the webinar and introduction of today's topic. So allow me just to stop sharing my screen and you can proceed.
Thank you. So as mentioned, part of this webinar is to promote our upcoming special MDPI issue on genetic engineering in agriculture, second edition. We welcome many different article formats, so we're hoping that some of you folks taking part would be willing to either submit or hopefully read some of these articles. Looking very much forward to it. Please visit the MDPI website if you want to learn more or see when the deadlines are. But Big part of why we're meeting today is we have some excellent science to meet new people and make connections. So very, very much looking forward to this. So just to give a little bit of context for why this topic is broadly of interest. So as we know, our human population is continuing to increase. We have a growing need for more agricultural outputs. So this includes food from both plant and animal sources, what we call feed, broadly defined as feeding plants or plant products to animals, which we usually then eat the animals. Fiber, this would include plant products such as cotton or linen, paper. We also make fiber products from various animals. So increasing this productivity, as well as decreasing losses, would both aid in the goal of improved overall yields. So my background is in plants, so this is going to be a little bit plant-centric from this point on. So for plants in particular, many agricultural losses are from biotic factors. So insect damage, disease, weed competition, as well as abiotic factors. So climate change, drought stress, colder winters, hotter summers. All these are factors that are decreasing what we currently get from our agricultural outputs. So how do we deal with this? Well, one approach is genetic engineering. So this is one way that we can try and reduce losses. And it's really interesting. So genetically engineered crops are a very newcomer to how humans have been changing plants for millennia. But interestingly, the development and rapid uptake of engineered crops shows that we can get desired outcomes. Some of our early pioneer engineered crops were herbicide-resistant or pest-resistant or both. So the main players currently are corn, cotton, and soy, along with other ones that are coming on board. And these were very successful. These were our foundational genetically engineered crops. And since then, we've had a whole bunch of additional plant types and traits that have been actually brought to market or are in process. So some newcomers, we have virus-resistant papaya that's grown in the United States, mostly in Hawaii. We have non-browning apple grown in the United States and Canada. This was engineered to be non-browning and reduce food waste. And the most recent addition that I know of is pink-fleshed pineapple, which was supposed to be brought to market in 2020, a bit unfortunate timing there. But these are RNAi, and they're marketed as pink because it's a novelty. So it's just for fun, which is really interesting. So this whole array of new engineered crops and animals are all in development. So it's really interesting to see now that we have the technology and we can do things, what will we do? So we're going to see some of that today with our speakers. So we've already had a short introduction. Just wanted to introduce everybody again. So presenting first is Professor Bernie Wone from the University of South Dakota, who's going to talk about advancing plant genome editing. So some of this work is very cutting edge and some is proprietary, so will not be in the final recording. So folks, if you're listening to this later, there are going to be some gaps. That's okay. It means what we're hearing about today is fantastic and brand new. Next, we're going to hear from graduate student Ivy Lynn at Yale University. It's going to tell us about application of CRISPR-Cas9 to citrus trees. And our third and final speaker is Sebastian Kosiova, an independent researcher from Binomica Labs and an innovator who's going to talk about flowers from fiction, adventures in ornamental biodesign. So it's a fantastic group. Thank you very much to everybody who has agreed to take part. I'm excited to get it started. Thank you to both our speakers and our audience. So just as a reminder, there's going to be time after each presentation for questions and answers. You can just type those into the chat here. This is going to be great. And then there'll be time for more questions and discussion at the very end of the webinar as well. So if everyone could please join me in welcoming our first speaker, Dr. Wone. Thank you very much.
Well, first of all, I want to thank the organizers and specifically the chair for inviting me to present some of our work that's happening currently in our lab right now. And as she stressed, I'm really grateful for her to give an excellent introduction, and that's sort of most of the part of our work. And so my title of the talk is really sort of developing a sort of a new strategy to edit plants or to express large numbers of genes in any particular plant. And so I'll go ahead and get started here. All right, as the chair mentioned, that in terms of the plant genetic engineering, it's needed based on that we are having a very fluctuating climate. We have more mouths to feed and whatnot, and of course, genetic engineering is required. But besides generating desirable traits, another thing we need to also know is using genetic engineering to understand gene function in any plant. And that's one of the sort of goals of plant genetic engineering, to discover novel genes that may actually work well in certain environments. And of course, to generate desirable traits, traits, that is a higher yield or more antioxidants, or more ROSs, you name it. Plant genetic engineering is required for plant-based production of recombinant proteins. And we actually saw the use of plants specifically in tobacco during the COVID times, where the tobacco was used to generate recombinant proteins for vaccine development. So we do need to have better plant-based production systems and be able to express different types of recombinant proteins. And of course, last but not least, plant genetic engineering is to increase biotic and abiotic resistance. And given the conditions of our climate these days, fluctuating climates, really degraded habitats and whatnot, plant genetic engineering is required. But for those who are in the field, definitely know the drawbacks, or why plant genetic engineering has lagged behind the biomedical field, is pretty much there's a literal and physical barrier, and that is a plant cell wall. And so that has kind of kept the plant world moving forward with more advanced developments. Specifically, genetic transformation in plants is really a two-step process: delivery of the genetic cargo, and then eventually the expression of the generated transformed plants. Conventional methods used are the agrobacterium to introduce the gene of interest via the recombinant TI plasmid. That's very often used, and then also the gene gun. There are some other ones, but these are your sort of main ways to genetically transform plants. And then once you genetically transform the cells or the groups of cells, then you have to co-culture them. So that's the regeneration step of the whole process of getting genetic transformation in plants. And then you have to select for it and eventually repeat the process until you get your desired trait that be expressed 100% in terms of the transgene you're interested in. In terms of genetic transformation, these two steps each has their bottleneck. Delivery, and then also the tissue culture step. The tissue culture step, for those who worked in this, know how labor intensive and frustrating it is in terms of getting this done. In terms of delivery, that we actually kind of passed that in terms of overcoming that barrier, and that is the use of nanomaterials. Nanomaterials, you'll see nowadays in a lot of publications about different types of nanomaterials being used to deliver genes of interest, whether it be entire constructs or even RNAi, you name it. And it's nice that you can have all kinds of different nanomaterials. It could be a particle, it could be a nanolipid particle, it could be even micelles, gold plates, carbon nanotubes, well known about the whole thing with the single wall carbon nanotubes. You can do other materials. So, and the nice thing about nanomaterials is that you can pretty much make any size you want, in terms of the size exclusion of letting things in the plant cell is between 5 and 20 nanometers. So we can deliver genetic material via nanomediate delivery. And the advantages are pretty much, it avoids a lot of the stuff that we see in agrobacterium. It avoids cell damage from biolistics, from gene gun. It can avoid host limitations because agrobacterium has a limitation to certain plants it can actually infect. And also you can carry different macromolecules. And again, it doesn't matter what type of nanomaterial you're talking about. It could be magnetic, it could be metal-based, silicon-based, clay, carbon nanotubes, you name it. And the delivery of the nanocomplex that is carrying the genetic information can be done by co-culturing in a petri dish, in a suspension. It can be done shot in by also biolistics. It can be injection via needle, needleless and or needle, spraying, and also PEG transformation. So the delivery of the nanocomplex in terms of the nanomaterial and the gene of interest there's no limitation. And the results are, in general, nanomediate delivery of targeted genes most of it, unfortunately, is it's transient expression. It's rarely its stable transformation. And that's where kind of leads me to this talk is really nano-enabled transformation, that it's a transformed DNA remains in sort of in a separate state from the genomic DNA, which is good and bad. So for example, some of our work, we actually express genes of interest, such as the EGFY gene brought in by the biocompatible nanohydroxyapatite nanomaterial. It'll bring it in, cross the barriers, and then brought into the nucleus, and then the plasmid expresses the mRNA, and then the mRNA produces the protein of interest. But this is short term. And so this pretty much gets to sort of the meat of the problem, is that we can use any nanomaterial to carry our genetic information in, or genetic cargo, but the problem is that it's not a long term. So for example, if you look at this particular plant cell here, here's the genomic DNA, here's the nucleus, and let's say this is the plasmid to deliver via some nanomaterial, and then to express the protein, let's say this is betylene, the Ruby reporter, and it would express it once it's brought in, but for maximally maybe 3 or 4 days, and then it goes away. So it's transient expression in terms of nano-enabled transformation, which is good and bad, because now you don't have the protein of interest anymore. And if you want to edit any cells with a Cas9 protein, that short duration may not really edit enough cells to actually cause any changes. So if you want to increase the expression of that, then you need to add more of the nanocomplex. So this, then you're risking problems with, overdosing, or even impacting the host, also other organisms, because now you're introducing more and more of the nanocomplex into the cell and perhaps into the environment overall. And then so, again, a lot of these challenges I talk about is not mutually exclusive. They're all connected. And then because of low, the trans expression, you get low expression of the target genes. And so they're connected. And if you want to increase the expression, you need to put more nanocomplexes, then it can risk into too much of the toxic material being built up in the cell, and therefore causing cell growth problems and whatnot. And then another problem I see with the challenges of nano transformation is that you introduce DNA and it's not passed on because it's transient, all right? And that the plasmid nanocarrier complexes cannot replicate. So it's short term, that's it. I might be wrong, but please correct me. I think all nano-enabled transformation is transient. And if it's reported as stable, you should really look into that. And also another issue with nano-enabled transformation, because of the short-term expression, low expression, that you can't select for transformants. There you can't put a marker there to select did I edit the cells or not. Or the plantlets or the cells. You can't do that. And then also another issue is that the scalability. Can you bring up, in terms of manufacturing large amounts of nano material to bring to the agricultural industry? It seems like you can, but at the same time, it depends on how this stuff is manufactured. So that is an issue. And it also brings into the problems of regulatory concerns, because now we are already introducing a lot of nanomaterials into the environment. Can we introduce more, or should we introduce more? And this brings up me to talking about, for example, those microplastics. Microplastics are found everywhere now, in us, in me, in all of you guys. And so that's a concern. So do we want to introduce more nanomaterial? You ask me, my opinion, I would be very cautious. So ultimately, all of these are concerns with the nano-enabled transformation. They have many positive aspects of it, but at the same time, there are many limitations. So what's currently done to overcome these limitations, specifically a longer expression of the genes of interest, or better or higher expression? And so, of course, we can look at the stuff that's being done by Gelvin's lab, where they're using the VRD2 mutant to actually, again, express a gene of interest to edit the cell. But again, this transformation is it's a transient transformation. It's not stable, so it's a short term. Yes, you do not introduce this gene of interest into the genome, and at the same time, it's transient. And also, we're still dependent on agrobacterium, which has many issues as well. Another possible sort of overcoming the limitations and challenges of low expression and more stable expression is probably the closest we can get to is the work done by the Slocken lab of the Donald Danforth Plant Science Center, where they're using rice pong transposase to insert genes of interest. And they actually have shown that they can have targeted insertions of target genes, and it's shown to be work very well. And at the same time, the delivery of these rice poson pong transposase is again also dependent on agrobacterium, but they may be able to adapt it to using other delivery methods, which I can talk about here. So in terms of stable or long-term expression, these are what's out there right now in terms of the able to gain a more stable transformation for gene editing, you name it. What about other impacts that are sort of we can overcome? And so some of the other impacts I mentioned are risks to the environment, the organisms and whatnot, and therefore we can use more biocompatible or biodegradable nanomaterials. And this includes biopolymers. I mean, again, there's a whole host of those you can look at. Episomes, exosomes, liposomes, you name it. You can use clay. And our lab has also kind of looked into that and kind of got into the whole nano-complex formulation, and that's where we discover there are limits and challenges associated with nano-transformation. We use nanohydroxyapatite, which is a biocompatible nanomaterial, which is basically natural material found in teeth like us, and as well as also in our bones. So, and we demonstrated it's highly compatible in our more recent publications in plant nanobiology. So right now, what's there besides agrobacterium, virulent D2 mutants, and then rice pong transposase, maybe using more biocompatible? What else is there? And I've been searching the internet and talking to people, and I've really not come up with anything. So it kind of, because I work in both the animal world and the plant world, I started looking into the biomedical field. And so can we learn anything from the biomedical field? And absolutely you can. And one of the things I found out was that there is a thing called the animal episome, originally described and proposed by Pikachek, excuse me, I pronounced that name wrong, back in 1999, where they developed an animal episome that actually expresses, that is actually self-replicating, non-integrating, and into the animal cells. And one of the main features of this episode is here, this is one of their first ones, is that they have this a matrix attachment region. And then you'll notice too, they also have a selection capability, one selection gene compared to 2 selection genes in any plasmid. And then you can see the size is actually relatively small. So can we learn from the animal world? And yes, so I think we can, and that's where our work comes in. So I propose developing these things called synthetic plant episomes, or what I call simplisomes, to sort of be able to express genes pretty much persistently, but non-integrating. Same idea as the animal episome. And before we talk about what are synthetic plant episomes, what are episomes in general? In general, episomes are really small circular DNA elements that does not, or it can or cannot be incorporated into the genome, and depending on how it's designed. So these episomes are pretty much analogous to plasmids that we find in prokaryotes. Episomes generally are referred to in the eukaryotes, such as plants or animals, but episomes generally are not present in the eukaryotic world. If they do, they're probably derived from transposons and or viral type types of episomes. So again, they are small, sort of separate circular DNA elements, which can or cannot be integrated into the genome, sort of in vivo, or I call implanta. That is, you can actually see it and express in the living plant and not kill the plant and track it. And this is the one developed by the Yuan et al. Group, the Xiaohan's group in Oak Ridge National Laboratory, and they used a GFY gene that you can express and actually see under UV light. It's very characteristic. So a plant under UV light autofluoresces to red, but if it has that expression EGFIP UV, you can see blobs of green being expressed, and you can see that nicely to the roots of it showing GFP via the UV light. So you can track them. You don't have to even look under a microscope, so they're non-invasive. And of course, the other one is the Ruby one, which is basically producing the protein called betylene, which is a natural plant compound or dye and found in, for example, fruits such as dragon fruits, for those who eat those, they're bright red and whatnot, and produces that bright ruby color. To produce that, you need 3 genes, and he et al. Back in 2020 made a construct to express all 3 genes as an operon, where these genes are linked by self-cleaving peptides, and they actually demonstrated nicely that this particular report is, again, non-invasive and can track over time. So in terms of our work in progress, and then we're gonna, again, I don't have a lot to results to talk about, but actually, hopefully we'll have some in a couple of days. Unfortunately, not at the moment. Most of this work is being led off by my postdoc, Madhavi, and in terms of the whole process of assembling different fragments to assemble into a full episome. So we were able to assemble an episome about 76 kb in terms of the size of the episome. We call this the ESM1 because it uses the synthetic matrix attachment region and also expressing the ruby in terms of that. And then we assembled it, and then we actually transformed it into bacteria, and then we extracted the plasmid, and then we checked in terms of the size of the episome, and it did replicated and expressed in bacteria very well. And this is using, again, one reporter now for both in plant cells and animal cells. So it works well in bacteria. We haven't learned if it works well in plant cells or not. But we found our target. It actually expresses nicely right where it should be. So this is a 10 kb ladder, and you'll see that here's the 3 kb band, there's a 10 kb band, and our target rests right between the 6 kb and 8 kb. And then so we also ran a longer time to see better separation. Again, we see our target there in terms of assembled episome. So expressed nicely right between the 6 kb band and the 8 kb band. But very interestingly, a very high molecular weight DNA band showed up around 30 kb. As you can see, it shows up, and this shows up only after transformation in E. coli. And so we were just stumped and just digging into literature, and we hypothesized that I think that these are really concatenated episomes. And this has been reported back in 1970s in plasmids from bacteria. And we need to confirm that. So even though we have an assembled episome and we tried to transform it into plants, it didn't work the first time around. So we started looking at our design again, and we discovered some sort of problems associated. And so we had to redesign and go back. And now we solved the assembly portion. Now we have to solve the why wasn't expressing. And we figured that out, I mean, with our last iterations in terms of the design here. So right now we're testing a 5 kb episome in terms of the what we're doing right now. Then this one is a smaller version. It expresses the EGIF-UV gene. We're testing it in tobacco, wheat, soybean, and hybrid popular. And you can see that we can assemble it, but we ran into problems, not an issue of assembling, but more why it wasn't expressing in plant cells. And I think we discovered why in terms of why it wasn't expressing. So again, in terms of summary, I think our solution to the issue of non-persistent and low expression and non-replicating issues of all the plant transformation techniques that's out there, our synthetic plant episomes are designed to be non-integrating, self-replicating, and capable of persistently modifying their cells and their differentiated progeny, meaning that these episomes, if it expresses and replicates, it will be passed on to the future generations. And I understand up to 100 generations or more. So it's persistent, and you can stop that too if you wanted to. With that, I want to thank the funders, specifically a funding from the National Science Foundation EAGLE grant. They're funding this research here in terms of the development and sort of proof of concept of a synthetic plant episome. And then I want to also thank Bayer Crop Science for sort of getting some, not the specific work that we're doing right now, but past work that kind of led us to this journey here. And of course, I want to thank my lab for hard work. My other half, I call her the plant whisperer because she can pretty much make anything grow. And then the rest of my team, Madhavi, Tanishka, Nancy, and Chloe, they're all integral in helping develop this synthetic plant episomes. So with that, I'm happy to take questions.
Thank you, Dr. Von, for your presentation. I will invite you to check the Q&A section. You can find it somewhere on the bottom of the screen in the middle.
Oh,
When
okay.
you open, you can read them and answer to them.
Okay, so I guess what kind of risk? So there are actually first questions. How can, excuse me, how can we increase rice production through genetic engineering technology, specifically in Bangladesh? Well, to address that question is probably in Bangladesh, I would assume more issues of water or flooding. I'm not sure, but I can try to address that. But you can probably, if you find genes that tolerate flooding and whatnot, may help with that. In terms of I'm not sure what do you mean by rice production, increasing the yield or survival or what? I'm not sure what you're asking there. So if you can further maybe specify what you're asking. The other question is, what kind of risk is nano-enabled transformation? What are the risks of nano materials? Nano materials depends on what you use. So, and keep in mind that nano materials, some of the nanomaterials are very persistent and they persist in the environment and will not degrade. I mean, specifically, I know, for example, the single wall carbon nanotubes, basically they do not break down. So if it's carrying, let's say, a plasmid of interest, it can pretty much get into anything, even non-target host plants, because it may get into the environment. If it persists, then something might pick it up. Bacteria might pick it up, or another plant might pick it up. So that might be an issue there. So there's a lot of risk in terms of nanomaterials, making sure if you use any, that it's biocompatible and it breaks down, it degrades. And so the question of increasing yield, so it depends on, you can find a gene that, or transcription factor that can increase the yield. So that could be relatively easy done. And in terms of really finding which gene triggers higher yields and at the same time doesn't have an effect on growth, because oftentimes you have yield issues with growth issues. So you have to kind of balance the two. You can't just have one. And do plants have episomes? What is your confident regarding transformation is? What is your confident regarding the transformation? Well, the plants normally do not have episomes, as I expressed. Episomes are generally found in, if they do, it would be either virally derived or from transposons. So not to my knowledge, I'm not aware of any plant episomes. Okay, so that's naturally they're not there. So we're developing something that's totally synthetic.
Sorry for interrupting you. If we do not have other questions for Dr. Von, at the moment we can proceed with a second presentation. And if somebody of you remember anything to ask Dr. Von, you can leave the question again in Q&A section, so we will read them again at the end of the presentation. So now I'm inviting Miss Ivy Lin, and please, you can proceed.
Hi everyone, my name is Jenny Ivy Lin, and I'm in the Irish lab from Yale University. So first of all, I want to thank the organizing committee to give me the opportunity to be here and share what our lab has been doing, working with citrus for the past decade or so. So I'm going to give a very quick background about CRISPR-Cas9. Okay. So gene editing is very powerful now because of CRISPR-Cas9. And these components are first discovered in 2005, and very soon afterwards, it's been harnessed for genome editing in 2012. So Jennifer Donna and Emmanuel Charpentier, they basically simplify these components into a two-component system by fusing the RNAs into a single guide RNA. And this would enable Cas9 enzyme to able to target specific sites in the genome. And this got them Nobel Prize in Chemistry in 2020. And later on, Feng Zhang and George Church, they applied this technology into human cells and proved it works. And so since then, people are working on different species and trying to use CRISPR-Cas9 to edit different species, genomes of different species. So when the DNA is cut, the DNA repair system is very prone to errors, and this will frequently introduce a mutation into that site, either an insertion or a deletion. So then it's very easy for us to create premature stop codon to knock out that gene, or then we can fine-tune in the promoter region to have different expression level of that gene. Or sometimes we would even introduce 2 guide RNAs, and then we can potentially create a bigger deletion in the genome. So CRISPR-Cas9 is an easy, accurate, and effective method to alter the genome, and virtually it can really target any sequence in the genome. So with CRISPR-Cas9, now scientists have way more research that we can do. For example, like we can have targeted medicine that can tailor to individual patient and really tailor to their needs. And we can increase biofuels, for example, decreasing the content of lignin or like increasing the biosynthesis ability. And also with the climate change and all that, there are more disease and the disease are spreading even faster. So as plant biologists, we are really trying to have the disease-resistant crops for the field. And of course, there will be new industrial products like biopolymers or like any kind of like we can engineer the bacteria to yield a big amount of high-value chemicals for us. With that said, we also have some challenges. Like we are still not sure how safe this technology is, and is it ethical for us to edit all this genome? And once we have these crops that are disease-resistant but being edited, what are the regulations? How acceptable would the consumers be? So there's a lot of regulatory challenges that we are facing. So here I want to switch gear a little bit to talk about citrus. So there are some challenges we are facing when working with citrus. First, it's very high heterozygosity. A fun fact about citrus is that all the commercial cultivar that you see, like pomelo, mandarin, lime, lemon, grapefruit, oranges, sweet oranges, they all only from 4 ancestors, which means they are hybrids of hybrids of hybrids of hybrids. So it makes it very difficult when we are trying to work with one specific gene. For example, in our lab, we use Carrizo sea change as our lab rat. It is a hybrid of sweet orange and Poncirus. So when we are trying to study one gene, we need to know, okay, how many copies of this gene we have in the genome, and how different are each of the parental genome. Then we can like really narrow down to what we're working on. So that is for sure a challenge. And also citrus has really long generation time from one seed to one seed. If the condition is perfect weather, good amount of nutrient and all that, it takes a lot of time, about 8 to 9 years, which is very not friendly to a grad student. But then like we know that Arabidopsis only takes about 3 months. So citrus is hard to work with. And another, as Dr. Wong mentioned, the transformation is very difficult. So with CRISPR, at least we can tackle the high heterozygosity problem. So with CRISPR, in citrus world, we are able to try to investigate disease resistance genes. For example, there's a deadly disease. It's called Huanglongbing, HLB, also known as citrus greening disease. it has wiped out 80% of citrus production in Florida, and because it's transmitted by insects, so it will get to California very soon. So with CRISPR, we are able to try to identify some genes, and then maybe we can enhance the citrus resistance to this bacteria. And then we're also able to study horticultural traits. So if you know, citrus plant, most of them have thorns, and thorns are not a good trait per se, because then you would hurt the fruit when they're growing, or it's just very dangerous because you would be stabbed by them. So we also study these traits. In the lab, we have a citrus tree that we completely transform their thorns into branches, and this is due to CRISPR. And of course, down the road, we will be able to study fruit quality. So working with citrus, there are some steps that we are really trying to focus and optimize them. Of course, we need to optimize the CRISPR construct for it to be suitable for citrus. And then we really need to increase the transformation efficiency to be able to have more transformant. And then once we have these thousands of plants, then we need to validate what are their editing levels, which is a better mutant than others. So first, I will start with how we optimize the CRISPR construct. So since 2013, people first introduced CRISPR-Cas9 into Arabidopsis, and after that, everyone work with plants are trying to adopt it from that. And because citrus and Arabidopsis are closely related genera within the order of Malvidae, so we continue keeping the YAO promoter from Arabidopsis and driving the Cas9 cassette. And then we swap the hygromycin resistant into condomycin because it works better in citrus. And we also introduce a GFP protein here because then we can have a dual selection marker. And then the single guide RNA is driven by RNA polymerase 3 U6 promoter. So then here I'm showing an example of we knocking out PDS gene, which is phytoin desaturase gene, and this gene is required for chlorophyll biosynthesis. So if we knock out the mutant will be albino. So here I'm showing different degree of the mutant. Like the very left one, you are seeing some white dots around the leaf margin. And the middle one, you are seeing some spots. And then E, we're seeing more white than green, which means that it's a more successful mutant. But then, of course, we will have events that we can generate like pure PDS mutant. As you can see, it's completely white. And since we introduce a GFP marker, like this, you can easily see that this is growing GFP. So it is very easy for us to select the mutant when they don't have the strong phenotype as PDS mutant. And then we are also seeing that heat stress help increase the Cas9 activity. So here on the left-hand side, it's a tree that did not go through heat stress. So the plant is edited. As you can see, the green is relatively pale on the top. But after heat stress, the new growth are completely white. So then now we are able to have a very efficient CRISPR construct, and we are able to increase Cas9 activity to a desirable degree. So after that, then we are able to create double mutants. We put another guide RNA in. Here I'm demonstrating with TI1 and TI2, which is thorn identity 1 and thorn identity 2. So as I mentioned before, citrus plants have thorns, and these two genes are controlling the outgrowth of thorns. So on the left is an untransformed citrus, and on the right is the edited plant for TI1 and TI2 double genes. So that in this plant, all the thorns are transformed into branches. So that's why we're seeing a bushier plant compared to the wild type. So of course, we are not satisfied only being able to knock out 2 genes. We want to knock out more. So here we're showing that this is traditional method with single guide RNA array. So for example, if we are trying to knock out 4 genes, each gene will have a promoter, in this case, Arabidopsis U6 promoter, driving the guide RNA, then forming these 4 guide RNA arrays. And we are trying to replace it with a tRNA array. So then all the guide RNA are linked together, and together they are only being driven by 1 promoter. So this would make the construct a lot smaller, and it would be easier to introduce to the plant. And we showed that the editing efficiency is about the same between single guide RNA array and the tRNA array cassette. Okay. And of course, we want to know what's the best combination. So our lab has been playing around with different promoter, different terminator, and to see what's the best combination for citrus. And here I'm just showing one of the figure that we play around with different promoter of Cas9 and different terminator. And it is for sure showing that some combination is better than the others. Okay. So these are what we have done to optimize the CRISPR construct for citrus. And then now the obstacle is how we can transform into citrus plant. So as Dr. Wong mentioned, tissue culture is very time and labor intensive. For in citrus, we grow the seedling in the test tube for about a month in the dark. And then these epicodial of etiolated seedlings were used. We chop them into pieces about 1 inch long. And then we culture them with agrobacteria for about 10 minutes. And then we put them into the co-culture media for 3 days in the dark. Then after that, we will move them to regeneration media. And this from this step on, we have the rifampicin and cefotaxime to repress agrobacteria. And then, so then this regeneration media would increase the callus formation, and also new shoots would growing up from the callus. So I mentioned that we introduced a GFP marker. So it's very easy for us to see which young shoots are successfully transformed with this construct. And then once we select them, we will put them under the light on the elongation media for them to grow, for the stem to grow longer and stronger. And once they are tall enough, we would chop them down, put them on rooting media, and roots will form. And once the roots are healthy, we would put them into soil. So then after they be in the soil, we would put them into growth chamber to go through heat stress cycle. And once they're happy, we would put them into the greenhouse so they can grow as tall as they want. And this whole procedure takes about 8 months. And all the steps in between, we need to change them onto new plates every 2 to 3 weeks. So even though it has very efficient recovery of gene-edited plants, it's very labor intensive and very costly. So then we try to develop implantar transformation method. Basically, we are growing the seeds just directly into the soil. And once they are tall enough, we chop their apical bud, and then side bud will grow up. We put the cotton on the top, which is soaked of agrobacterium for a day, and then we remove them and put condomizing on the cut side. So then they would be selected in this way. So here is a photo showing that GFP is the edited plant and the red one are non-edited plant. Those are autofluorescence. So a sad news is that the editing is not as efficient, but then the turnover rate is very fast. It's about 2 to 3 months. So these are the two protocols that we have been using in our lab, and we are still constantly trying to improve these protocols. So now we have thousands of plants. How do we know which are the good mutants? So here we are proposing an affordable whole genome sequencing protocol. So for example, I have 50 plants. I collect the leaves from these 50 plants. I put them into one tube. I extract the whole genomic DNA from these plants. And then we add an enzyme, which is called MSPJ1. This enzyme is really special because it only targets the heterochromatin region and digests that part. And then we will remove the digested fragment and send our wanted part into sequencing. So here I'm showing the results. The top line is a whole genome sequence coverage, which if you just collect the genomic DNA and send it for sequencing as normal, you can see the coverage is pretty much the same across the whole genome. But with our protocol, which is digested by the enzyme, you can see that the euchromatin area is very highly upregulated. The coverage is a lot higher compared to the heterochromatin region, as you can see here. Okay, and this is like proved. Like you see, like the transcript are in this region and in this region, and then the heterochromatins are where there's a lot of transposable elements. So give you guys an example. So for example, this vector 245 is targeting 4 different positions. And with this whole genome sequencing, we are able to know that position 1 and position 3, they are perfect, have really fairly high editing score in this population, but position 4 only have 2.5. So we are trying to find each plant that has like perfect edit, right? Then we will go through Sanger sequencing. We sequence a bunch of plants at their position 4, and then we found those plants that are perfectly edited at position 4, and then we go back to check position 1, position 2, and position 3. So in this way, we are able to prioritize which plant we do Sanger sequencing first, and this would save us a lot of time and effort. So this is just another demonstration of this is selected. So if we start from where position 4 is already good edited, then we go back to other position, then we have really high chance to get a perfect mutant. But if it just randomly selected, it's really hard to have a good mutant that all 4 positions are edited. Okay, so in this talk, gene editing is a productive approach to mutate specific gene in citrus, and it's very fast, cheap, and precise, and it's a lot easier than conventional breeding. So gene editing obviates this issue associated with GMO because it doesn't require the insertion of genetic information from other species. And a good news is that in the US, the USDA stated in March 2018 that gene-edited plants that closely resemble their conventionally bred counterparts will not be under regulatory review. And this, over the years, all this regulation has been improved. So with that, I'd like to thank my PI, Vivian Irish. She's such a great mentor, and thank all the former and current lab members, and also our collaborators within Yale and outside of Yale. And with also the grant, USDA, CRB, and NSF. So I'll take question if there's any. Thank you.
Miss Amy, thank you very much for your presentation. And Miss Ivy, I'm sorry. And I see that we have 4 questions. 2 are in Q&A section, and I see other 2 in chat. So you can proceed with them.
Okay. So first question in the Q&A, can CRISPR-Cas9 accelerate the development of seedless citrus variety compared to conventional breeding? So now in our lab, to do transformation, we need seeds. So it's not very, it's not possible to work with seedless varieties. We could do propagation of them, but then afterwards, CRISPR would be really difficult to do. We are trying to generate our mutant with implanta, and then hopefully 8 years later, when they have fruits, the seeds, we grow them, and then we can apply another agrobacteria on them so we can have double mutant in that sense. So maybe not, but who knows? Science does improve very, very fast. The second question is, what are the major target genes in citrus that have been successfully edited using CRISPR-Cas9 for disease resistance? Okay, so there's a gene that's called POP21. It's published this year, and it's been proved to be really resistant to, I'm not sure if it's HLB or it's just like citrus canker, but then there are some genes that we know to be important to combat this disease. So a lot of different labs, we are trying to combat HLB from different angles. Like some labs are trying to do gene editing as our lab, and some labs are trying to do like antibiotics, or some study the insect side and try to combat this disease together, basically. The other question is, how low is the gene editing efficiency of CRISPR using in implanta transformation? As you can see on the last photo, if you are really lucky, then probably 20%, but most of the time you are not that lucky. So like the GFP positive plants would be like 5 to 10. And then from those plants, we would test their edited level. Okay, so those are the questions from Q&A in the chat.
Yeah, there
Okay.
are 2 in chat.
Okay. Can we use CRISPR-Cas9 in organism other than plants? Yes. So it's actually first tested in human cell. And so basically any organism, as long as you have genome, CRISPR can work. Is CRISPR-Cas9 the only knocking out gene or also doing transformation? I'm not sure if I understand this question completely. Basically, CRISPR-Cas9 is making mutation, either insertion or deletion, and then ideally in that gene, you would create a premature stop codon, so this gene would not be functional. Has the injection method of agrobacterium been tried for citrus transformation? Yes, it has been tried, and it's not the most efficient in our lab. And I know other lab has been trying to do like protoplast transformation and all sorts of different tissue transformation. So yeah, everyone's trying. I hope I addressed to all the questions, and if there's more, you guys can type it in the Q&A. Thank you.
Thank you, Miss Lin, once again. I believe you answered to all questions, and if anybody has more questions, you can leave them in Q&A section, and we will check them again at the end. Now I will invite Mr. Koisioba for his presentation, and please, you can proceed.
Thank you so much. That was a really cool series of talks. Hi, everybody. My name's Sebastian. I want to be a flower designer one day. That's my ultimate goal in life. I've been working with plants for the last 20 years, have been running a one-man contract research organization out of my home lab for the last decade. I focus strictly in biosafety level 1 work with bacteria and plants, and my research focus is in the homeotic gene expression towards novel flower morphologies and exploring that landscape through both rational and irrational design. And I'll explain what those two are in a bit. Okay, so quick grounding exercise. So we are here, specifically here. This is seen from the Voyager spacecraft in a couple months before I was born in 1990. And this is everyone. This is the Earth. So this Earth has wonderful things like water. It's a wet rock. It's got lots of dry land. And it's got some funky green stuff that I love. So what happened on this rock is unique to our planet, as far as we know. It's an elaboration of life. So this is not to scale by any stretch of the imagination and is a bit eukaryocentric. Bacteria tend to dominate this, but this is a common ancestry tree of life, where all of life came from a theoretical common ancestor and then elaborated into what we have today. And I'm really interested in the green stuff, the plants on the right. So I love plants. When I was little, I saw this broccoli at a grocery store, and I was just floored. I couldn't understand it. I didn't know what I was looking at. And it was right next to normal broccoli. So I told my mom, I'm just like, how can we eat the regular broccoli? We have to eat this. What is this? So I had no idea what I was looking at the time. And many, many years later, there was a publication on understanding the Romanesco broccoli and the Fibonacci spiral of gene expression that leads to these beautiful morphologies. So it's like a living display of mathematics, which just captivated me. Plants are incredible. Here's General Sherman, an 83-meter tall tree that it takes like 42 children in a circle to hug it. It's enormous. It moves tons of water up and down the trunk of its tree, of the tree each day, using essentially nanotechnology, exploiting capillary forces, using cellulose and little very engineered veins to move upward water essentially for free. We have so much to learn, from building composites to manufacturing to industry, medicines, and agriculture, as we've spoken of. But I love flowers. I love flowers so much. They're the most inspirational thing for me. They're the most morphologically diverse tissue type in all of living things in our planet. And so this is a rose. I'm not a huge fan of roses, but when people think of flowers, they tend to default to roses. When we're in middle school and in high school, we're taught the anatomy of a flower, right? We have these petals and sepals and anthers and ovules. We have all of these things, and it's very easy to discern what is what, and it's very cleanly displayed for educational purposes. But flowers are actually like really weird. So on the left, we have Fritillaria melangensis, which is the snakehead lily. And it's one of the few plants in nature whose petals have a checkerboard pattern that's actually like clearly defined squares of alternating anthocyanin production and lack of production, even though there's no tissue types that morphologically divide those pieces. On the right, we have the fairy twin orchids that have these like funky little tails, and they just look so strange. Where is this on that flower model, right? They get even more stranger. Right, especially in orchids. They're the most diverse of all flowering plants. That thing in the middle is a fused petal. You can see the tiny sepals on the bottom. And then we have this style with strange feather-like protrusions. The anatomy gets very complicated. And I love that. It's just so morphologically free to express, especially from an artistic perspective. They also have structural color. So on the right, we have a blue morpho butterfly from the deep in the Amazon. And on the left, we have a begonia, who when you shine light in a specific angle reflects the same way that butterfly does. So the butterfly has no blue pigment in it. It's actually reflecting due to the structure of the keratin that's building up the wing. And on the left, the begonia, it's actually the thylakoid stacks. So the chloroplasts themselves are so tightly arranged that they reflect light in a specific way, causing this birefringence and iridescence. So flowers are beautiful, flowers are diverse. I'm obsessed with it, so much so that I had to drop out of university for family and financial reasons, but I was addicted to research and I wanted to do more. So slowly but surely, I taught myself a little bit of electrical engineering to repair used equipment on eBay, flipped them, kept the best for myself, and this is my lab today. So this is in a nook in my house. It has a laminar flow hood, gene gun, PCR machine, et cetera, et cetera. All of this is used and broken equipment that I fixed myself. Fancy stuff, but realizing later that you don't need the fancy stuff to do good biology. And I'm working on making educational tools so people can build their own tools and explore the living world. Okay, so rational design. I call it, we kind of sort of know what we're doing. So it actually, this whole journey started with a bit of spite. So this is the Applause Blue Rose from Suntory, the Japanese whiskey company that commissioned Florigen, a plant engineering startup, to make a world's first blue rose. And this was published. It was deregulated. It went for sale, and they were selling it for $25 a head. Now, my schooling isn't advanced, but in middle school, I was taught what blue was, and I feel like this is purple, a cool lavender at best. So I feeling deeply insulted by the lack of understanding of primary colors. I went on a quest to make a blue rose. So diving into the literature, I found that there are pigments that make blue, but there's also proteins that make blue. And I was way more familiar doing protein expression than metabolic engineering. And so I started diving into chromoproteins, proteins that have a color in daylight, not just UV absorption, but daylight absorption. And so I found some blue ones that were kind of crappy. I did some mutagenic PCR to make them more expressible, more soluble, and now I had a blue. So it's like, okay, this is blue, not purple, this is blue. So going through that notion, I said, okay, let's get other colors. So what about red? Here's E4CP from an anemone. Beautiful, beautiful red. There's no UV here besides the small amounts of UV light coming in through my window in daylight. Then I found some yellows, mutagenized a bunch of yellows. I found 14 to be my favorite yellow, the most yellowest yellow I could find. So the next I needed a darkening agent. And I'll get to that in a sec. So I found that Rhizobium etli produces melanin through a gene called MEL-A in the presence of copper. And I expressed it in E. coli and put it on plates with copper, and it started producing melanin. Cool. So now we have a blue, so we have a cyan, I guess, a magenta, a yellow, and a black. Well, that's the CMYK color spectrum. That's how printers use subtractive color spaces to make all the colors that we see when we print out paper in color. So by having just 3 colors and a darkening agent, you can make the perception of any color you'd like. So I started cobbling together a set of chromoproteins. Shameless plug, I sell them in my web store. The entire rainbow except for green. Green's actually really hard to find as a chromoprotein that's not metal binding. I'd like something that's just purely amino acid sequences and no cofactors. But I'm working on that. I have a kind of a crappy green. Hopefully it'll be out next year. Yeah, so I have all these colors, and I thought, okay, what happens when you mix them? So here is extracted chromoproteins that you mix 20 microliters at a time in a gradient from blue to red, and yellow to red. And in between, you can actually see different colors. You can see peaches and lilacs and greens. So I thought, okay, this is actually coming. This is culminating in something. This is a color gamut that I can play with. So I thought, what if we put all of these on a single plasmid, right? The blue, the pink, the yellow, and the polyphenol oxidase to make melanin, right? To make plant version of melanin. And have it on a single plasmid, and then have students compete to modifying the promoters and expression machinery so that you can have like the Pantone color of the year reflected in a plant, right? So I'm working on a cool competition to get students involved in flower design this way. So exploring some of this, here is a disabled tobacco mosaic virus where the capsid protein coding sequence was replaced with the green fluorescent protein. This is a plasmid produced in like the 1980s by the Losomanov. I'm sorry, I keep mispronouncing that name. That wonderful lab that did a lot of TMV research. And you can actually see it spreading through the plant, but it doesn't have the machinery to turn into viral particles that can then exit the plant. So it's just a roving, self-replicating mRNA construct. Some of my chromoproteins were actually used in making colorful plants. So my friend Kelsey did this wonderful work taking the TS purple, optimizing it for lettuce, using this really intense lettuce promoter. You do need a lot to express chromoproteins, a lot of protein expression, especially because the vacuole occupies so much volume of the cell that you can't, you only see like a small thin line of it as the light passes through it. So you don't have that depth to really reach saturation. So either like vacuolar tags or higher and higher expression levels to be able to see it compared to fluorescent proteins, which are kind of cheating because you get more photons out. Yeah, so this is being expressed in plants. A bunch of my friends are doing it in a whole bunch of different plants, getting various degrees of expression. Really excited about the blue stuff. Okay, so beets. Beets offer color in plants fantastically. They're just beta lanes that Dr. Wan explained is utilized in a circuit called Ruby. I don't need to go into too much detail about Ruby, but like Dr. Wan said, it's a single transcript containing 3 coding sequences that each have an enzyme chain that turns tyrosine into beet juice, essentially. And it works really well. So you can, this is tobacco, you transform it. I used a marker-free transformation, so I just regenerated after exposure to agrobacterium, chasing the red, right? So no kanamycin, no antibiotics used. I started subculturing the red bits, cutting out the green bits. They started growing. I had to keep on getting rid of the green bits. Sometimes some of the shoots started silencing for reasons that are beyond me. It's just like, okay, now we're green. Cool. Some of the mutants were really, really fascinating. This is a temporary a mutant where half of the meristems that led to this leaf were transgenic and the other half were wild type, causing this like half moon phenotype. This was all transient. It reverted back to stable, and because I ferreted out a lot of the chimeric tissue, but I would love to come back to this and actually get a stable half and half phenotype. Okay, Ruby roots are insane. They're so vibrant. This is an unedited photo. My phone was like glitching out because the magenta was saturating the camera. And the resulting tobacco plant was beautiful and bronze colored as the green comes through the red. So lots of like really aesthetically interesting sculptural motifs that could be explored. And the stem looks just fantastic, especially through light. And here's an example of, in the same batch of plants, some had a lower expression due to probably positional effects. Some are extremely expressive, as the one on the left. And yeah, so that was color. That was some of the things that I've been exploring in terms of color production. But let's get a little bit into shape. So this is a very simplified version of the ABC, now E, ABCDE, and sometimes F flower model that's increasing in complexity of understanding how transcription factor units come together to signal different tissue specificities. So how petals elaborate, how the floral meristem starts to emerge, how plants transfer from the vegetative to the floral state is heavily researched, and we're understanding using Arabidopsis tremendous amounts of knowledge that can be then transferred into other plant models where there are variations and caveats, of course. So what's really fascinating is that these homeotic genes that are responsible for the ABC flower model also produce really interesting phenotypes when you knock out said transcription factor pairs, especially on the right. So on the left, we have wild type. You can see the standard Arabidopsis flower. Then you can start seeing ones that don't have petals, ones that are just infinite amounts of pollen-producing organs, and then the agamus mutant, which is nothing but petals, which is fascinating from a biocontainment standpoint because no pollen is being produced, right? So these are inherently sterile; they can't reproduce, but they make beautiful, beautiful art, right? And so here's the work of my friend Nick Desnoyer, who took Arabidopsis, and by stacking some of these homeotic mutations and expressing Ruby, he made an Arabidopsis rose. And he's working on thickening the stem and just really going crazy with what normally is a very, very fine flower, a tiny little flower, into something more significant and more impactful and visually stunning, all as educational tools. I'm really huge thanks to him for pushing this forward. Okay, so we also learned that the stacking of these mutations, dominant or recessive, or basically the zygosity of these mutations, can lead to really interesting emergent properties. You see here dashed in yellow different notches that are formed because of these crosses that are reliably forming. So we have yet to fully understand the pleiotropic effects of these singular mutations when you stack them together in a single plant. So truly endless forms, most beautiful. We are understanding that the silencing of certain transcription factors can actually extend the amount of time that a petal undergoes for cell division, such that you get more petal, right? So it takes longer for the plant to fully bloom, wild type above and then the silenced below. It takes longer to fully bloom, but the result due to the timing window is an expansion of the petal. So as long as you stay in that petal cell division, that cytokinin heavy state for longer, you get larger petals. We also know that because life is ironic and has little jokes, when you truncate certain transcription factors, you truncate the length of certain petals, right? Which was really fascinating. So there's multiple MADSBox and other binding sites within those transcription factors on the surface of those transcription factors that allow them to bind to several places that by truncating them, you have a smaller peptide that leads to less binding to a secondary site, for example. So on the left, you have wild type, and on the right, you have a shorter ligule. And certain species of plants actually use this to further elaborate their floral display for pollinator attraction and subsequently for human attraction, because we find this beautiful too. We can also mess things up when we have pairs that are very important, that when you silence one and not the other, you get the different morphologies. And I love this because, like, yes, we're kind of screwing up these flowers, but what we're further understanding is like the subtlety and the nuance of potential designs given all of these factors. So the plant looks normal if you have these two expressed. If you silence one and not the other, you get these really interesting divergences from the standard. We also know that microRNAs now play a really big role in the regulation of morphology in plants. There are microRNAs that block the TCP genes that can then result in the elaboration of waves within Arabidopsis. So you can make curly lettuce phenotypes of Arabidopsis by just editing and shutting down several TCP genes, right? So you can stack those mutants. You can do essential like large CRISPR array that you can knock those out all at once, and you get this very heavily wavy phenotypes, which then can be recapitulated through genome sequencing of curly lettuce. I bet you would find those similar type of knockdowns there. Especially the subtlety is what I'm most interested in, like the microRNA silencing of certain factors can lead to really, really subtle changes in the structure of the leaf, like we see on the figure A on the very bottom. So there's lots of nuance that we can play with. There's many, many moving parts, but that in those many moving parts are more paint for our palettes, more clay for our spinning wheel. We also have the ability to express certain variants of that transcription factor that can lead to more or less anthocyanin production in certain species, like here in Petunia, where you essentially almost have a dial. It's stepwise, but it's something. We can start playing around with these type of things. And the results of a lot of these morphological changes and metabolic engineering changes have become commercially available. Like here is the Arctic apples that after 3 minutes, when you cut them, one browns and one doesn't because of a knockout of the polyphenol oxidase gene that causes that browning. So now kids in middle school will actually eat their apples instead of throwing out because they're brown. Another one that hit the market that I love is the Norfolk Healthy Produce Purple Tomato. So they took genes from Snapdragon and put them into tomatoes, and now it makes an overwhelming amount of anthocyanins, which are rich antioxidants. They look cool. You can buy seeds. I think they're like $10 for a little packet. You can just grow them and cross them and have fun with it. And the last one that is shiny and new is from Light Bio, a company that made a true autoluminescent petunia. So the plant glows in the dark using the Lux genes from Panella stypticus, if I remember correctly. And they used a recycling gene that takes the oxoluciferin, the burned luciferin after the glow, and turns it into a Krebs intermediary to allow recycling of energy, causing sustained glow. Now, irrational design. So we talked about things that we kind of sort of know what we're doing. And now let's talk about some things that we have no clue what we're doing, right? So leaning into the noise. So it all started with Atoms for Peace. So after the atomic bomb dropped, we wanted to have civilian nuclear utility. And so they found that you can do this thing called atomic gardening, right? So on the right, we have this large slug of cobalt-60 that if you see it, you have to drop and run, as the instructions say. It's screamingly radioactive. But they found that you can actually make a circle around that cobalt slug and plant different plants in there and cause mutations, right? So this is called atomic gardening. And a lot of the processes involve high energy particles, like high energy physics, like gamma rays and fast neutrons that allow for double-stranded breaks. And if you are fortunate and the double-stranded break is in a wrapped up piece of DNA at that moment, you can cleave through multiple pieces at once, causing massive deletions, rearrangements, etc. Processes like this is how we got the ruby red grapefruit as one example. There's many others like it. Here's an example of a similar thing in Japan that's actively being pursued right now. It's claiming to be the fastest breeding process on the planet, where similarly you have a radiating area of cobalt-60 gamma rays that are coming from that slug, and it radiates out where you see trees in the outside and annual crops in the middle. And it's being used today. So ever since the 1950s, we're doing this today as well. There are other ones. So I spoke of neutron beams, gamma rays, similar mechanisms. Heavy ion beams can also do breaks like that. The gamma rays are nice because they can do essentially single-stranded breaks, right? Which could break a single nucleotide, and then in the presence of other mutagens could also be could cause a frame shift mutation. It can cause many different smaller mutations. And the company that was doing the gamma irradiation in a single year got all of these phenotypes for chrysanthemum by literally just placing cobalt in front of it, walking away, and then planting it later. So there's still a lot of untapped morphologies and cultivars that can be generated using something as crude as blast it with spicy light and wait for the plants to grow. Another mutagen is EMS, right? Ethyl methyl sulfonate, if I remember correctly. It is a known carcinogen. Many mutagens are carcinogens, so it's very dangerous to work with it, but it actually can do GC to AT swaps because of the mechanism of their process, and it occupies a space. It can bind to thiamine, causing, instead of a GC, it would have to be an AT once it's repaired. And so as a result, you can do nucleotide substitutions using this. The process of it is very simple. You expose the plantlets or the seeds to EMS, a concentration like 1% for 60 minutes, and then you regenerate and you get all kinds of different phenotypes. But it puts a tremendous risk to the researcher. It is a carcinogen. It has to be disposed of properly. There's so many reasons why not to use this routinely. But there is a mutagen that I've fallen in love with that I'm actively working with, the sun, right? So the sun produces, as a black body radiator, a full spectrum according to the Maxwell's equations in physics. And one of the rays that doesn't reach our floor because of the filtering of our atmosphere is UVC radiation. But we actively have UVC radiation bulbs, germicidal UVC, around 254 nanometers, that are capable of producing mutagenic levels of radiation with an off switch. And you can shroud it, and it's light. It can't penetrate through certain materials. And so UVC radiation works typically with pyrimidines. So it typically does TT dimerization. So if there are two T's near each other, it causes a cyclobutane bond, and it causes a kink in the phosphodiester backbone of DNA, which then can inhibit DNA polymerase. But life, of course, finds a way, and there are two main mechanisms of repair. One of them is nucleoside excision repair, where an enzyme that looks for that kink literally sits on it and then cuts it away, such that then DNA polymerase can come in and fix that broken bit. The other one is photoreactivation, where an enzyme, photolyase, sits on that kink and waits for a single photon of visible light to strike it, which is enough catalytic energy to break that cyclobutane bond, repairing that TT dimer. But if you keep things in the dark for a while, those lesions can be carried over, right? Can become heritable mutations. So I made a spicy light death ray, which is just a really tiny UVC light, and holding a reusable Petri dish with an Arduino. I wear proper PPE, of course. Always protect yourself against UV light, especially germicidal light. And I got really good results with E. coli. This is just 60 seconds. On the right is a negative control lawn. On the left is 60 seconds of exposure. I'm actually actively doing a process where I'm mutagenizing K12 every day for 30 days and then sequencing everything to see this like GIF of the rearrangements and mutations. Because pyrimidine substitutions and mutations are the common mutation type, but there are many other mutation types, including double-stranded breaks and deletions, just at a lesser, to a lesser extent. And I'd like to know what extent that is. Recently, which I'm so excited for, is I found out that caffeine is a mutagen at very high levels, right? So it enhances EMS mutagenesis, but there's also been papers that have shown that high concentrations, like near the solubility threshold, concentrations of caffeine in the presence of seeds can lead to heritable changes similar to EMS, including chromosomal aberrations, DNA polymerase inhibition, all kinds of different mutations that can be gleaned with this. And we drink coffee every day. So of all the mutagens you can think of, this is one of the safest. Now, what has been shown is that UVC mutations are synergistically amplified by caffeine. And so I'd like to have both of them together to see if I can actually get very, very profound mutations in my irrational design atomic gardening simulator without having to use the scary cancer juice that is EMS or other mutagenesis. Okay, so my canvas organism. So I talked a bit about rational and irrational design. What am I designing? So for a really long time, for like a decade, I've been looking for my canvas organism, the organism in which to paint on. And one summer, a couple summers ago, my mom went to a flowers market and like a roadside flower stall, and she saw a handwritten sign that says white petunia, $5. And she bought it. And she was like, hey, try this petunia, see if it works. And it was mostly a joke because I tried so many petunia cultivars and they were recalcitrant, hard to grow, non-disease hardy, et cetera, et cetera. I have a long checklist of things that I really want to see in a plant. And I tried it just to humor her, and it was the fastest regenerating plant I've ever worked with. I have 25 days from culture to flower. And as a flower designer, that's a great turnaround time. And like 35 days seed to seed. And it grows in falcon tubes really well. It's my canvas. You can inject Ruby with agrobacterium directly into the petals. And this is a clockwise time series over the course of 3 days. And you can see the Ruby expression is super bright. And then after 5 days, the flower falls off and resets with a new flower. So you have like the equivalent of Nicotiana benthamiana agroinfiltration, but every 5 days without growing new crops. So that's fantastic. I also have some mutants that I'm super excited for. Here's a crinkly phenotype. Of course, it's male sterile because of my luck, but it's beautiful and I've crossed it a bunch of times and I'm super excited to just have my own cultivar and play around with it. So I'm making all of these open source tools so that people can explore biotechnology and genetic engineering, hopefully towards making some really strange things. So here is the Dr. Seuss truffula tree on the right, and on the left is the Australian grass plant, which is not too far off. So playing around with some of those transcription factors that we talked about, or the CRISPR arrays that Ivy spoke of, we could potentially knock it down and make an actual truffula tree. My biggest source of inspiration is this book. So this is a book by Luigi Serafini called the Codex Zephyrinianum. And it's about a planet that doesn't exist, written in a language that doesn't exist, about plants that don't exist. And here are some of the plants that don't exist, but I really want them to exist. And if you really stare at them, some of the morphologies could be had. And so I would love to take my petunia and really, really depart from what a petunia is to some of these wild and wacky things, and hopefully inspire the next generation of researchers towards doing things that are creative and expressive and somehow, hopefully, ingratiating a sense of environmental stewardship in the process. So thank you so much. I'm a tiny green cell on all social media platforms, and I'm happy to take questions.
Thank you very much, Mr. Quesioba. I really enjoyed in your presentation also. And I would now invite you to open Q&A section. I think you have 6 questions, if I'm correct. And you can start one by one.
Okay. So Abdul Quddus says, if engineered floral sterility or alternating pollination traits escape into wild relatives, what could be the long-term ecological biodiversity consequences? Can you give some examples? Okay, that's going to take a bit to fully unpack. But so floral sterility in the very sense of it is not going to allow it to be transferred into the next generation. So it intrinsically is making something more safe because the pollen's not floating out there. Altered pollination traits. So a lot of so everything that I make, because of the regulations in the US, biosafety level 1 is not regulated, but the unlawful dispersion of GM organisms into the wild is very much regulated. And so everything that I make lives and dies in my lab unless a gallery that I show allows me to do so, to bring it to them. So in state, I can transfer around with it, but it can't be released, right? And so within the scope of my work, it won't be released anyway. But in the scope of deregulation, we would have to go through all of those to see whether or not it's safe, whether the mechanism of action, according to that 2018 change to the law, where the mechanism of action doesn't impart weediness, et cetera, et cetera. Like there's many traits that are a no-go or have to be field trialed to be deregulated. So before it would ever escape, we would have to go through deregulation, and it would just be a hard stop if there's something that would lead to ecological damage. But we can discuss more of that because that's a very, very large conversation on bioethics and beyond biocontainment. Okay, another one. I've benefited from your open source protocols. I have been an avid follower of yours. Thank you so much. I appreciate that. Do you have any recommendations starting point for optimizing UVC and caffeine dosage? Would love to mutagenize extra orchid protocorms. 1% caffeine and a lot of UVC. So like I start with 10 minutes, but honestly, like do a kill curve until you kill your protocorm, and then work backwards from that ceiling until you get something that's just barely surviving. And that's spicy enough to be able to inherit. Now, remember, when you expose, whatever morphologies you see in your M1, the first generation, is not going to be heritable necessarily. Those are just lesions. Those are scars on the plant. You want to actually make them self-pollinate and then screen their M2s, the generation of that plant, to see how the morphologies segregate, how those traits segregate, and if it's stable. Hey, Nick, very inspired talk. Curious what your take is on flower design as a form of art. Can one truly express themselves through genetic design? we don't have that much time to explain that. But yes, absolutely, as a canvas, genetics as a means of painting, as a means of sculpting, especially with flowers, because they're so morphologically infinitely diverse, you can actually get intentful expression, especially now that we're shifting from I don't know what I'm doing to I kind of know what I'm doing, right? So the most important thing about art is that it has to carry intent. So whatever you do, whatever sculpture it be it living or non-living, has to carry intent. So as long as intent is there, even a rudimentary expression of modifications towards that intent is significant enough to be called art. That's my personal perspective. What state do I live in? New York. Can flower dip be tried for crops that are recalcitrant to transformation like melon? So I don't have any experience in melon, but I highly recommend people to definitely try floral dip and all the other mechanisms that was discussed in this in these last two conversations. Specifically because we don't know until we try, and these negative results are so critical. And I strongly encourage people to publish their negative results. I have tried all of these floral dips, none of these worked. Such vital information for both the reproducibility of science, but also to save people time. Because if you really tried and you have a lot of evidence to support that this doesn't work, maybe that's not a tree worth barking up, right? Okay, that's all those questions from the Q&A.
Sorry for interrupting you. I think there are a few more questions in the Q&A section when you scroll a little bit upwards.
Oh, okay. Let me see. Oh, okay. Yeah, sorry. Sorry, I might have missed this. How do you express the ruby gene in tobacco? What transformation method did you use? So I used agrobacterium transformation, standard 35S double enhanced promoter for the expression cassette. And yeah, I just used standard tissue culture and the like boilerplate agrobacterium tobacco transformation method. So 1 milligram of BAP, 0.1 milligrams of NAA every 2 weeks, transfer until shoots form. But I wasn't using any antibiotic. I was chasing the red. So I had a lot of chimeric tissues that I then had to chop and reculture. I don't recommend you do that. I was just trying to prove a point. Okay. Could altering flower architecture genes improve pollinator attraction without compromising yield? Sure. Yeah. I mean, like so for example, there are pigments in the UV spectrum that we can't see that insects definitely can see. Like there are daisies that have bullseyes, but only can be visible in UV light. And by modifying those, you could potentially make your GM plants more attractive, right, to certain pollinators. But that comes with like ethical concerns and ecological concerns that need to be really carefully understood, because you don't want to be leading pollinators to a plant that isn't going to be successful. And modifying the ecosystem in that manner, I don't think that we've had large longitudinal studies on genetic engineering of plants and the pollinator preference over many, many years. So I would be very, very careful with that. But yes, you can definitely change pollinator attraction. Without compromising yield, I'm not sure. What's the potential consequences of manipulating self-incompatibility genes in crops like rice, wheat, and mustard? So I'm not a monocot guy. I mostly work in dicots. I focus more on like nightshades and mustards. So from a self-incompatibility genes. So I would love to see modifications for inducible self-incompatibility, right? As like potential kill switches for agricultural products. But that would be a really long discussion. If you want to message me on social media or send me an email, I'd be more than happy to answer. Okay. I think.
Yeah,
I hope that's everything.
I think you catch up with other questions
cool.
that you already answered. And I think there are a few more questions in chat. I will just check.
Somebody asked, what would you develop with $1 million grant? A national educational pipeline for making flower engineering accessible to middle school and high school students. Yeah. I think that the trickle down and the abstraction of these tools are capable enough to be able to be handled by very, very precocious 8th graders, but definitely high school students. And if anything, especially community colleges in those places that really, really need it, I would love to see more hands-on biotech. And there's something really profound about making a genetically engineered organism and physically touching it, as opposed to the poop bacteria on a plate, which is interesting intellectually, but not so interesting to engage with. So yeah, if I had $1 million, I would definitely make an educational pipeline.
Yeah, I think other questions are technical questions for my colleagues and myself.
Yeah. If there are, I don't see it in the chat. If you can just shout one out, I'd be happy to.
I think you answered to all. There is just one question from Nick that is a little bit broader than he asked you via Q&A section.
Yeah.
Do you,
Okay.
do you see it?
Yeah. Okay. I see the irrational versus rational. So Marcel Duchamp made the ready-made, right, which makes art out of an object that already exists. And so the irrational design could be semi-ready-made. There's room and debate from an art perspective as to whether or not that counts as intent. But again, that is a huge question that I would actually love to have a call with you, Nick, and discuss that and record it and publish it so we can have contrasting opinions on what it means to be intentful in rational and irrational design. Yeah. But I think you can. It's just complicated. Okay. Awesome. I think that's it for me. Thank you so much.
Thank you once again for presentation and for answering large number of questions. And now I would invite our chair, Dr. Amy Klocko, for closing word.
All right. Well, thank you very much, everybody. I thought these were 3 fantastic talks showcasing what tools do we have, what can we develop, how can we make them better, what can we do, which is really, it's amazing now. This is a global topic. We have a global audience here, so it's really exciting to see what can happen. So if there are any final questions from our audience, we didn't really get much into the ethics and bioregulation. That's probably for the best. It'd be a little more contentious now. But yes, I really like to thank everybody for joining in. If there are final questions, please do put them in the Q&A. We'll wait a couple minutes for that. Otherwise, I think we'll be closing this out. I know the final recording will be a little bit edited to get rid of some of the proprietary information from earlier. So if there are pauses or gaps, that's why. So.
Thank you, Dr. Klocko. I think nobody is writing more questions at the moment. So I think we can close our webinar. I will just say something for our attendees that is important for them because we are providing certificates of attendance for this webinar. I encourage you all to check SciForum platform to download your certificates. The link is already shared in chat multiple times. I think that you will be able to download certificates after the webinar is closed. If somebody has issues with downloading it, you can write us via email, but we already have your email, so I guess we will send it to you some follow-up emails. And if you met any issues for download of certificates, you can write us. That was all from my side. And if there are no more questions, I would like to thank once again to all our speakers. I also enjoyed myself in your presentations. I hope that also attendees enjoyed in your presentation and learn a lot from your research. As I said at the beginning, this webinar was created in aim to promote the topic that Dr. Klocko is leading. And I encourage all attendees to check the web page of the topic and read all published papers in the first edition. And I encourage you also to consider publishing your own research in second edition of the topic. Thank you once again, and I would like now to close the webinar.